Good morning, everyone, and welcome to the closing end of Latin XChem. Today, it is a great pleasure for me to introduce you to Professor Rudolph Marcus, who's going to give one of our plenary lectures today. So, Rudy Marcus, he's a Canadian-born American chemist who received the 1992 Nobel Prize in Chemistry for his work on the theory of electron transfer reactions in chemical systems. He studied at McGill University in Montreal, gaining a science degree, and then he stayed on to earn his PhD in 1946. He then joined the new postdoctoral program at the National Research Council in Ottawa. At that time, there were no theoretical chemists in Canada, and in 1949, he gained a postdoctoral research fellowship at the University of North Carolina, USA, to work with Professor O.K. Rice. In 1951, he became an assistant professor of chemistry at the Polytechnic Institute of Brooklyn, where he experimented with gas phase and solution reaction rates before turning to electron transfer reactions. During the 1960s, Marcus wound down his experimental research to concentrate on theory. In 1964, he joined the faculty of the University of Illinois, where he continued to study reaction dynamics. He spent the year of 1975-76 as a visiting professor at Oxford and Munich, and in 1978, he left Illinois for the California Institute of Technology as a professor of chemistry, and he has remained there since. The practical consequences of Marcus' theory on electron transfer, it really extends all over areas of chemistry, from photosynthesis in plants to chemiluminance, electrically conducting polymers, corrosion, the methodology of electrochemical synthesis and analysis, and much more that I'm not going to talk about today. I just want to share you before giving the microphone to Professor Marcus that it's almost 3 decades ago that he won the Nobel Prize. I happened to be a high school student back then, and I participated in the International Chemistry Olympiad. And they gave us this poster announcing his Nobel Prize, and there was a blue copper protein site there. Little did I know that later on I will be working on blue copper sites and thinking about electron transfer reactions in proteins. And I'm sharing these because many of our students, postdocs, and professors participating in Latin XChem, so many Latin American chemists were once inspired by chemistry at that stage of life when we were in high school. And so I want you to know, Professor Marcus, that you have been an inspiration for a lot of young chemists to go on and study these kind of things. So with that introduction, really, we want to thank you for being with us today. And it's a great pleasure to have you. Welcome.
Well, thank you very much. It's a real pleasure for me to meet with you all and to talk about some of the work that we've been involved in, that we found exciting, interesting, puzzling, just about every gamut of emotion you can imagine. In this talk, I'd like to make it also a bit historical, because the theory, this electron transfer theory, didn't just sort of arise from nothing. There are all sorts of things that preceded it, and its development involves a lot of almost coincidences, accidental things. I think that probably happens quite a bit in science, at least for some of the unusual things, because if the results are unanticipated, unexpected, there's a better chance that it's novel. If the results are just what you expect, then that's nice, but it's not necessarily producing new things. So I would like to make the talk a little personal and describe a few of the things that happened. Well, let me see if I can get this thing for me, the problem of electron transfers arose when I was in the library around 1955 or so. I was in the library, and I used to go to the library every day to look at the latest journals. And in those days, of course, it was before computers. And I happened to come across a symposium in the library. This is, again, unexpected. And this symposium was on various chemical reactions that involved isotopes. This was not so long after the war. And so there was a lot of isotopic research going on. And in fact, quite a few Nobel Prizes have been won for various aspects of using isotopes as tracers and other ways. And I happened to be just reading through some symposium that had occurred a couple of years earlier at the University of Notre Dame. And it was largely on what's called isotopic exchange reactions. These isotopes were available, radioactive isotopes were available in all sorts of materials because this was the age of atomic energy. And so a lot of chemists use the isotopes to trace chemical reactions. And one of these chemical reactions was isotopic exchange reactions. So here's an example of ferrous-ferric. The star designates trace amounts of a radioactive isotope of the iron. And when the reaction occurred, this radioactive isotope appeared in the ferric ion instead as a result of an electron transfer from one ion to the other. And by chemical separation, they were able to measure the rate of production of this particular isotope. And from that, they were able to calculate reaction rates. Now, there are many such reactions studied because there are many different kinds of metals and hence many different kinds of isotopic exchange reactions. And one interesting thing about these reactions that made them different from all other reactions was that essentially, apart from where this radioactive label was, these reactions, the products were the same as the reactants. That meant that one of the driving forces for a reaction, namely how downhill or uphill it is, was absent in these reactions. And sometimes when you can simplify things, in this case, getting rid of the driving force, when you can simplify things, there's a chance that you might have something that's easier to treat and where certain effects are more transparent. And in this symposium, it pointed out that if one looks at all of these electron transfer reactions that have been studied, there is one rule that emerged, and that is that the larger ions, ferrous are pretty small, but there's some large ions of other materials with various complexes attached to them. The larger the ion, the faster the rate. And this was faster not just by virtue of ion size, but this is, that's the disadvantage of having a phone connected to telephone. I was able to deactivate the telephone, but not the phone. All right. Well, anyways, I can hear this phone in the background. Yeah. These were the smaller ions were slower by orders of magnitude. If it had just been a matter of ion size, it would have been a factor of 2 or 3. But these are orders of magnitude smaller. And the question is why? So here's an experimental fact that demanded a theory. You know, so often in interesting theoretical work, there's some experiment which is unexplained, and then you develop a theory to explain it. And if things work out well, science has advanced that way. Well, anyways, the question is, how do you explain it? Well, one of the people at the symposium was Willard Libby, who later won the Nobel Prize for radiocarbon dating. Libby suggested an explanation for why the smaller ions were slower in electron transfer than the large one by orders of magnitude. And his idea was this, that if you look at an ion and the solvent around it, the solvent is highly organized around the ion. And the smaller the ion, the more the sort of organization, the more the molecules of the solvent are oriented towards it. Now, when you transfer a charge, then you have to change all of that, those oriented molecules, make them less oriented if you're going from a large charge to a smaller charge. So the orientations of the water molecules, say solvent molecules, around an ion depend very much on its charge. So that means in an electron transfer, ultimately, there's a big change in the ordering of the water molecules around the ion. And Libby pointed out, well, if you have a smaller ion, it's going to be much more interactive with the solvent molecules. It has much bigger effect, and there will be a bigger barrier to reaction, because if you're producing products in the wrong environment, the smaller the ions, the more wrong the environment, so to speak. So that way he was able to point out that with small reactants, you have a slower electron transfer reaction. And basically what he applied was what's called the Frank-Condon principle, whereby when an electron jumps, the nuclei don't have time to move, so they're in that original environment, and that was the wrong environment for the products. Well, I was very excited when I saw that, because I'd already done some theoretical work in unimolecular reaction rate theory, and this was entirely new to me, applying the Frank-Condon principle to a chemical reaction. So I got pretty excited about it. And then the more I thought about it, though, something felt wrong. You know, often before you prove something, you go by feeling. You sense that it might be wrong or something, depending on one's intuition. And then I realized what was wrong was that Libby's explanation, although correct in applying the Frank-Condon principle, made an error he actually violated the law of conservation of energy, because certainly when the electron jumps, then immediately the two ions have a wrong solvent environment for the around them, and so they're high energy, and the smaller the ions, the higher the energy, the greater the barrier. But where did that energy come from? And actually what had happened was that here are great scientists, occasionally great scientists do make errors, had violated the law of conservation of energy. So the question was, how could one take Libby's outstanding idea of applying the Frank-Condon principle to these ions and make it work in a way that one didn't violate the law of conservation of energy? So that's how the electron transfer theory got started. And I realized that what had to happen is that before the electron transfer occurred, you had to have a change of the solvent environment around the reactants to something that was maybe more in between what the ion was before electron transfer and what it would be after electron transfer. So here's here's a sort of a crude depiction. The molecules, these triangles are water molecules, are more oriented around the ferric ion than the ferrous ion. And in the products, it's the reverse. So somehow you have to have some intermediate where the orientations are more similar. And so that was the essence of the idea. And the question is, how do you calculate this, this quantity in between? It's not as simple as what I've written there. You have ions, you have, in terms of electrostatics, you have the polarized solvent, the average orient, the dipoles are sort of oriented towards the ions and more oriented towards the more charged ion than the other ion. So how do you handle that? This is sort of a situation where all of the orientations are not those that are in equilibrium with the reactants or the products. Here you have equilibrium orientations, and here in the products, you have equilibrium orientations, but it had to go through some sort of an intermediate where not just the inner layer, but all around it, the medium was not that which was sort of in equilibration with the ions. And so the problem was, how do you treat systems like that? And the first treatment I gave, which was in 1956, was in terms of dielectric polarization theory. In other words, the ions interact with the sort of average dipoles of the solvent. And this picture here indicates that what one had to do was to develop the theory in a way that one had fluctuations of that dielectric polarization that are away from the equilibrium dielectric polarization around each ion. So how do you handle that? Well, you have to get into the electrostatics, or what you might call non-equilibrium dielectric polarization systems. And here is the connection with my previous work. You know, very often scientific works bear some relation to each other, even if they're in very different fields. And in my case, I'd worked for several years before this, 54 and 55, on the theory of what was called polyelectrolytes. And that's where you have a long chain molecule with charges attached to it, and you have counter charges around that. And how do you calculate the free energy of these systems? So for that, I did a lot of reading on electrostatics, a tremendous amount, and found a book that I really liked that made things clear and physical in the way it was handling things. So I'd done a lot of electrostatic calculations, but these were all of the kind of calculations where the solvent polarization is in equilibrium with all of the charges. And what we needed for the electron transfer was to have it where the electrostatics, where it's not in equilibrium, where you have a fluctuation. And of course, when you think of it, all chemical reactions really involve fluctuations of one kind or another, whether there's fluctuations of bond lengths so that a bond can break, or fluctuations in the polarization of the solvent. And so the key thing was, how do you extend electrostatics so as to treat what's called non-equilibrium dielectric polarization? And I found a way of doing that because of the detailed background I'd built up in polyelectrolytes. And so then emerged with a picture of reactants in terms of the free energy as a function of some reaction coordinate, which I won't discuss here, but there are various things that we did there. And so here are the reactants free energy, here's the products free energy. And you go from here to here in the reaction. The polarization around the reactants is dielectric is different there from there. You have an intermediate dielectric polarization here. So you need to be able to calculate the free energy where you have fluctuations of the dielectric polarization. And that I learned how to do in part because of the electrostatic background that I built up. And then next, let's see, did I skip the slide here? I guess that's all right. This is there's a quantity called lambda, the reorganization, which appears, it'll appear in the next slide, appears in the free energy barrier. And that lambda, there's delta G0, there's, this is lambda, that's sort of mathematically what lambda is. Let me just go ahead here. This is the expression that we ended up with for the rate constant in terms of a known pre-exponential factor and this reorganization and the standard free energy reaction. So this is the equation that emerged, but with expressions given for lambda. Here we are with the expressions again. And there are many consequences of that equation. One is that if you look at what's called a cross reaction, in other words, supposing you have two isotopic exchange reactions, but you mix them. You take the oxidant of one and the reductant of the other, and you're looking now at an electron transfer reaction that, unlike the isotopic exchange one, had finite delta G0. And anyways, the rate of the cross reaction we showed, it came out to be related to the rates of the isotopic exchange, the equilibrium. So this is one of the consequences. There are quite a few consequences. This was one of them for the electron transfer theory. And the advantage of it is that, often when you're comparing theory and experiment, sometimes there are adjustable parameters. Well, it's not very satisfying to sort of work with adjustable parameters. So if you can somehow relate one experimental quantity to another that doesn't have any adjustable parameters in it, then that's sort of more stringent test and a more satisfying test, the experiment. And this was one of the consequences that came out of this kind of analysis. These are the rate constants of the isotopic exchange. This is the rate constant of the cross reaction. In other words, reaction between one reductant and one oxidant where they're not the same quantity, different isotopes. This is the equilibrium constant. This is a known function of them. So this was one of the quite a few consequences that came out of the theory. Now, here's a test of the theory. First of all, there are many tests involving isotopic exchange reactions, and things worked out pretty well using that equation that I showed you. But one surprising thing occurred. Often in research, if you get something started, then somebody might take the equation, say if you develop an equation, and apply it for something that wasn't intended for, it wasn't derived for entirely. And so in this case, Lewis and Hu back in '84 considered this reaction here. This was a methyl cation transfer through aryl sulfonates from one to the other, and they had many substituents there. So they were able to look at the observed rate and the calculated rate, calculated by that cross relation, and you see things work perfectly well. And that was a big surprise because that cross relation was derived for electron transfers, but here is something that wasn't an electron transfer, and yet it worked. And that'll have implications for the work I'll be talking later. Here's another one where Wolf and Mitchell did a theoretical, quantum mechanical theoretical calculation of an SN2 reaction where an anion attacks a methyl iodide or other groups there. And they made a lot of quantum mechanical calculations for this. And here's a comparison of theory and experiment. The points are the calculated and the straight line is the points are the calculated quantum mechanically and the straight line is the theoretical result. So here is an example of an equation that had been developed for electron transfer reactions, but was applied by others, not by me, by others, to reactions that had nothing to do with electron transfers, and yet the aspects of it worked. In this case, that relation between the rate constants that worked. So sometimes there are unexpected consequences of theory, and that was one of them in my case. But it'll be related to some biological motor work that I'll describe later on. When I saw some of the results that have been obtained, particularly some experimental results where they applied the cross relation to them and it seemed to work, I wondered, well, the cross relation then should have some more general derivation, which isn't tied to electron transfer. And so I did some very elementary, you might call them quantum calculations. I considered this and I used the bond energy bond order model that Linus Pauling was involved in for calculating the energy as a function of the bond orders of this bond and the newly forming other bond. And these are the expressions. And anyways, when you do that and carry through some of the algebra, you get results that actually turn out to be, it's a different function, and one can understand that. But it turned out to be very similar in over much of the region to the function for the barrier that had been derived for electron transfer. These were all energy calculations. The other was free energy, but the ideas are similar. So that was an eye-opener that you could derive something for one set of reactions, and it might turn out to work for another set, but of course the details will probably be different. In other words, when one uses the cross relation, one didn't have ionic radii in it and distance and dielectric constant, optical dielectric constant that was in the theoretical expression for electron transfer. It had other things in it, but the final expression didn't have those specific things in it. It just had the rate constants and the equilibrium constant. So if you can derive an expression where it doesn't depend on the specifics of the process, then you have a chance that the expression might be more general than for the particular reactions it was derived for. And that was the case here. And all of this will be related later to the work on the biological motors that we're describing. One of the predictions of the theory, there are a number of predictions of theory. One was the cross relation. Another was what was called the inverted effect. You know, normally, if you have a chemical reaction, and if you have a standard free energy of the reaction, and it becomes more negative, typically that reaction is then faster. And in the theory, if you remember the expression we had 1 plus delta G over lambda all squared. In the theory, when delta G0 becomes very, very negative, then that quadratic expression, which goes through 0 at some point when delta G0 is minus lambda, becomes large again. In other words, the reaction becomes more and more downhill, but the reaction becomes slower. That was a consequence of the theory, and readily understood in terms of those free energy curves if we took the time to go through them. So it was physically understood, but it was an unusual prediction. And many people tested that prediction. And for the longest time, the results were negative. And then finally, in 1984, in this slide, an organic chemist, Gerhard Klaas, and a physical chemist, Miller, and their student got together and made compounds where you kept the donor separated, quite separated from the acceptor, separated by this organic bridge here, and then they finally observed the inverted effect. So after 25 years or so, approximately, well, more than that, there was evidence of this unusual prediction of the inverted effect. Since then, there have been many examples of the inverted effect, but it took 25 years. And in part, this worked because the reactants were kept separated by this rigid bridge, so that there was no chance of some kind of shortcut, another mechanism coming in. It had to be an electron transfer mechanism. So this was a pioneering paper of Miller et al. And changed the way many people look at things forever. And in fact, the Nobel Committee cited this prediction of the inverted effect in 25 years or so before it was predicted. Now, this inverted effect, that when it's very, very downhill, the reaction may be slower. That's been invoked in photosynthesis, whereby a wasteful, very downhill reaction doesn't occur or occurs with very small probability, presumably because of this inverted effect. So it has consequences beyond being an interesting oddity. You know, in theory, life really thrives by experiments. We rely so heavily on experiments to guide us on interesting problems to work on, and experiments that try to predict and what have you. And one of the predictions of the theory was that if you do an electrochemical experiment and you measure the log of the current versus the sort of potential drop across the, let's say, electrochemical cell, then for the longest time, the plots that were found there experimentally were linear. They're called Butler-Volmer plots, one particular case, and that was widespread. And then in the theory, we'd predicted there would be curvature, just like you saw curvature in the preceding PowerPoint. And many electrochemists try to get evidence to see whether they could find that curvature or not. And most of the results were, or all of them essentially, were unsuccessful because to observe it, you had to go through a region where the reaction was very fast. But when the reaction was very fast at the electrode, the diffusion of the reactants to the electrode was slow, and so you couldn't observe it. All you observe would be that slow diffusion of the reactants when you're working with those reactants. So for many decades, or several decades, electrochemists were unsuccessful in getting the predicted curvature of the log of current versus potential across the electrochemical cell. And then finally, Chitsey did a special preparation where he looked at the electrochemistry of a ferrocene ion and had a carbon bridge here with sulfur thiol groups attached to a gold electrode. So here the reactant was, let me see if I can get that back. No. Oh, no, wrong way. Yeah. Yeah, so Chitsey then made that layer, and now there is no slow diffusion of the reactant. So he was able to observe, and here's the observation, and you can see the curvature. Normally, you just see a straight line in systems where you didn't have that that situation where the reactant was bound, and hence there wasn't a slow diffusion. And so this was the first experiment which on the electrochemistry, which confirmed the effect. And this had major consequences for the interpretation of experiments. Nowadays, it comes up in nanoparticles where because of this curvature, it's much more accessible to reactants. The diffusion plays less of a role, and so you can get this effect, the curvature in principle anyway, in the electrochemistry of nanoparticles. Let's see now. Yeah. So in summary, for that quadratic expression, for the free energy barrier, oh my, I move this thing. The applications, there's to electron transfer, both an experiment, and there have been quite a few theoretical calculations now of that that quadratic expression, which have actually confirmed quantum mechanical, statistical mechanical calculus, which would confirm the expression. And then there is the application that I had nothing to do with, where Sutton did some experiments and quantum mechanics theorists did the calculations and found that quadratic worked pretty well. Then there was the methyl cation transfers. That was an experimental result that where it was applied by Lewis. And again, it was something I didn't anticipate. And now Jim Mayer has studied proton-coupled electron transfers, and the inverted effect has been found for it. And Hamish Schiffer has done a lot of the theoretical work on proton-coupled electron transfers, and the quadratic relation plays a role there. And now here's the introduction to what we're going to consider. We're going to apply that quadratic expression to a very different thing, namely where you have nucleotides going into a cavity in a biological motor, and you break a bunch of hydrogen bonds and you form new hydrogen bonds. So we're going to treat that by the quadratic relation too. That's not something which is rigorously derived or anything like that. It's just done as an intuitive step. And so I'll spend the rest of my time on that. You know, I took off my watch and I lost my track of what the time is. So
you are okay still. You
okay.
talking. Yeah, you still have another 10-12 minutes or so.
Okay, all right, good. So let's look at now the application of the electron transfer theory to the biological system. Well, first of all, there's the photosynthetic reaction center. Here's a bilayer membrane, and this is one of the applications of theory. You shine light on the photosynthetic reaction. Well, you shine light on the system, and eventually the energy is transferred to a special pair in the photosynthetic reaction. And whoops, daisy. Then an electron is transferred to the other side of the membrane, and all sorts of things happen. So electron transfers are involved in there. And part of the, also in the membrane, is this enzyme here, this ATPase, which takes ADP, an inorganic phosphate, and makes ATP, which is used by biological systems for as sort of energy source. And accompanying that is also a proton transfer. So we're going to apply it to this ADP, or really the reverse thing. We're going to apply it. We're going to apply that quadratic expression, and I'll just spend a little time on that. All right, so electron transfer theory then enters into the various steps here and enters into this, well, not as an electron transfer, but as another kind of transfer into this ATPase here. By the way, I think there are 5 Nobel Prizes involved here in various aspects of this diagram here. Here is this ATPase that we're going to be talking about. And, it's absolutely ingenious, just ingenious. Here you have this layer, this bilayer, and in the bilayer is this F1, F0 ATP synthase. And protons come in from one side, and then there's a proton transfer reaction here, and that causes this quantity here, oops-a daisy, that causes this quantity here to rotate, and then that causes this to rotate, that causes this whole thing to rotate, and because of the asymmetric nature of them, causes them to open and close clefts during the rotation. And during that reaction, it's possible for ADP and inorganic phosphate, PI, to go into a cleft and then stay for a while, and the rotation causes a compression, which causes the formation of ATP. And then the rotation also causes another cleft to open up, and the ATP comes out. There's absolutely, nature is absolutely ingenious in this. And of course, these rotor systems appear in other things beside this ATPase here. There are other biological motors where it also appears. And so there have been quite a few single molecule studies, not on the whole quantity, but if you snip this off and you look at the reactions going on inside the other part, then there have been a lot of single molecule studies of those systems. And that's where we came in, or really Professor Sander Vogen-Katsko came in. He had been an expert in treating these single molecule studies of other systems, quantum dots, and here were all these single molecule studies of biological systems. So perhaps one might be able to apply information about one to the other, or the way of thinking about one to the way of thinking about the other. And he got heavily involved in this. And the work that I described is largely collaboration with him, and wouldn't occur without him. All right, how do you describe a biological motor? How do you bring in the mechanical part into the chemical part? That's the whole key thing. And what in that motor, what you find from the single molecule studies, that there are certain stable angles of rotation. And the system goes from one stable angle of rotation to another stable angle, to another stable angle of rotation. And during the course of doing that, various clefts open up and close and permit chemical reactions to occur and quantities to go in and come out. How do you combine mechanical motion and chemistry in order to have a theoretical expression for that? And so what we did was that around each dwell, the dwell before you go to the next dwell, you have fluctuations, and I've denoted those by W for the first dwell, and then you have fluctuations in the next dwell. So by bringing those quantities into the free energy of reaction, here I brought it into the free energy reaction. This is the free energy reaction. By bringing them in there, and also in, before, to get to the intersection region, you need to have some work term there. By bringing them in, that was a way of combining mechanical work terms, which are really you can observe them actually in flux. You can observe these fluctuations of these in these single molecule systems. That was a way of bringing them into this whole formalism. So the idea then to combine the combination of chemical reactions and the mechanical motion, we use the fluctuations about each of the dwell points to bring in the mechanical part, and combined it with the delta Z0, which is the chemical part. This delta Z0 is the chemical part. That's the mechanical part. Then that way we were able to apply this expression. Now, of course, using this quadratic expression, it wasn't derived rigorously for this, but in virtue of the success on simpler systems of the quadratic expression for systems for which it had been not derived, then we decided to try it for this motor system. And you get expressions, and this is at one dwell point, the reactance is the stablest. The next dwell point, the product is the stablest. And anyways, you can discuss this. And we published this work in PNAS in 2015. I'll just show you one of the results. This is a comparison of theory and experiment with no adjustable parameters. And this is for binding of ATP. This is one of the steps, and release of ATP. And the dots are the various dots are the experimental quantities, and the lines are the theoretical quantities. So there are no adjustable parameters there. And this was something we published in 2016. So where one can, and sometimes one can, others one wouldn't, where one can, it's always good if one can test relations where you don't have adjustable parameters. Unfortunately, that's not always possible. It was possible in this case. Now, this is an example of two of the steps that have been measured and how they depend on angle. One of the steps is independent of angle. The reverse step, the hydrolysis, is dependent on angle. And in the previous slide that I showed you, the slope there would be in between those two. That's the way it is, and that's the way it should be for certain quantities. But when the transition state is either very close to the reactants or is very close in nature to the products, you'll get this kind of behavior. And so this is something that we're investigating now.
We have 4 minutes left.
Okay, I'll need 3 minutes. All right. Now, here's an experiment, controlled rotation, where they looked at an ATP that had been fluorescence attached, so it would light up, or if the ATP went into the this cleft of this motor, then it was rigid, so to speak, and it could fluoresce, and if, otherwise it wouldn't. So you could follow the rate of ATP going in and coming out, and ADP going in and coming out by the fluorescence, either it was on or off, and what have you. And here are the raw data obtained by Dachi and coworker Noji coworkers. And the lines here are, I mean, this looks like a terrible mess, doesn't it? I mean, if I looked at that, I would not try to work on it. But if you notice, there are areas of concentration and areas less concentration. And so the authors were able to sort of divide things up and could attribute it, the different parts to one of the clefts, another part to another cleft, another part to another cleft, and open or closed, what have you. This is not something I tried to describe here, but the main thing is you get data which in a sense look like a mess. But then when you go through analysis of the data, as Shander and another co-worker, Hannes, have done, then, I'll show you, I think. let me go ahead to that. Yeah, then you get this sort of result here, where we can understand these are different concentrations, and one is ADP coming out, and the other is ATP going in. And the main thing is that the curves are close to each other, and that we can understand the differences, why this red triangle curve tends to be on that side, and the blue thing is on that side. They're slightly different processes, so on. But the main thing is that sometimes you have data, the single molecule data, which looks horrible. I mean, if I were just looking at myself, I would never do it. But where it turns out by a very careful analysis of the single molecule data, you can extract all sorts of interesting information. So these are coworkers. Without Sandor, this work would never get off the ground. And Riccardo Mattuti is playing an important role in relating structure to the various events that are happening. He's the one who knows the most about the quantum mechanical calculations. Is invaluable. And some of the other co-workers like Wei Chen Chen and Kai Yu are working on problems very different from this. So these are three students, former students of Sandor, and they're doing important work in the group. And over the years, we've had a number of agencies that have supported us, and science should proceed by love alone, but you really need some financial support too. At least we found we do. So thank you very much, and I'd be glad to entertain any questions if there are. Thank you.
Thank you very much for the talk, Professor Marcus. If you want to ask a question to Professor Marcus, if you're in the audience in Zoom, you can just raise your hand and ask directly. If you're following us on the YouTube channel of Latinx Chem, you can write your questions in the chat and we will read them here. So while we get questions, I would like to ask a question. I would like to ask you, so when we're thinking about metalloenzymes in biology, like really complex ones like cytochrome C oxidase, or the ones you showed on the oxygen evolving complex, a lot of times the protein sets up the site and optimizes it to minimize the reorganization energy of the metal center.
Right.
And sometimes that's kind of a challenge when we're trying to do biomimetics and do that with the smaller molecules. What will be your thoughts on that challenge? Like, what are your thoughts on this?
Well, of course, there are a number of different systems where people are trying to entertain nature, like in the photosynthetic system. They have an acceptor and a donor and separated by various things. So nature, of course, has had billions of years to produce these things. And we as individuals have just had a matter of less than a century to produce things, typically. So nature's sort of evolved over a period in a way to optimize various operations. So I think it's a great credit if you can make synthetic systems that somehow get at the essence of the nature system and get results that are somewhat similar to nature. I mean, that's one way, a very good way of trying to understand what nature is doing, which of course is a subject of interest to many of us. So I think it's, that's a very useful thing to do, and it forces one too to look more closely at the biological system to see what is there specifically about it that makes it work in the function that it's doing.
Yeah, and the protein makes a difference, right?
Oh yes, I mean, it's well known in the protein That
So,
if you change one amino acid for another, sometimes even quite distant, 20 angstroms away, that can change the whole structure thing and have a major effect on the reaction rate, even though it's far from the reactive site.
yes. Well, so we have some questions here in the audience. I want to ask Antonio Romero has a question. Can you, yeah, you can unmute yourself. Great.
Thank you. An amazing conference, Dr. Marcus. I have always been your fan since I heard about the fascinating electron transfer theory. I have applied it on my computational studies in free radicals. Science will be not the same without your transcendental contributions, Dr. Marcus. I want to ask, what will be the future for electron transfer theory? Thank you.
I don't know, because for one thing, there's been this application and extension to this proton-coupled electron transfer. Well, you know, I didn't foresee that. So clearly, I'm not a very good predictor of things. In other words, some of the best things are really the unexpected, the unusual. It's not immediately obvious that there's a connection, but that was one of them. And I mean, electron transfers, of course, are ubiquitous. They occur in so many things. You know, like solar's energy conversion and electrochemical cells and what have you. So all of those remain to be explored. And there are sort of new systems that come along, you know, like the perovskites and what have you, and that affects treatment of solar energy. So electron transfer theory in its present form is ubiquitous, but we've seen some examples like proton coupled electron transfer and like that atom transfer, where some of the basic ideas seem to carry over, but of course there are some key differences, and there may be more of those that I personally haven't thought about, but maybe new applications. You know, there have been a number of applications which arose out of new experiments that weren't anticipated. So it's not going to be a purely theoretical thing. Sometimes one's very fortunate in making some prediction and that, but often it's the experimentalists who develop new results and in some cases apply theories for which they weren't designed, and it works. So that sort of causes that theory to sort of be looked at in a more fundamental way. And why is it that works? And what can you do to maybe do a theory that has a lot in common with that theory, but is really more appropriate for the particular reaction? But yeah.
Thank you. We are running out of time. We just have time for one more question. We have Juan Pablo Senocian in the audience. Can you ask your question real quick, Juan Pablo?
So, thank you for a great overview of electron transfer reactions, Professor Marcus. I was just wondering, looking at the Atpase rotating molecule, it reminded me of the problem of hindered internal rotors. And I was wondering if any of these models, like the Pitzer-Wynn or model, have been tried on this problem.
I think not for the biological motors, but for some of the other motors for which a Nobel Prize was won. I think things like that hindered rotation, and also the flexibility and geometrical changes that go on plays a prominent role. In this particular case here, the conformational changes are really more connected with the opening and closing of certain clefts as a result of various asymmetric groups sort of pressing against each other. So I think there's a lot to be done there in terms of understanding that aspect of the motors, that mechanical aspect of the motors. But I think the closest to what you're suggesting is in some of those synthetic motors.
Great. So
thank you.
unfortunately, we just run out of time. So thank you very much, Professor Marcus, for this great talk. Thank you for your time to Latinx Chem. It's been a pleasure to have you.